Hi folks, and welcome to my podcast. Today I'm going to talk about friends or foe, the microbes in our gut, and how they are influencing current trends in nutritional science and perhaps are poised to become one of the fifth pillars in the alphabet soup of nutritional assessment. So first and foremost, what are we talking about here? Microbes and the microbial environment in our gut are basically like their own ecosystem. There's bacteria, protozoa, fungi, archaea, and all kinds of viruses living together inside us. And that's kind of gross, but it's also kind of cool. They outnumber us by an astronomical amount. And they're often changing. For the most part, they go along their merry little way and we don't even notice their existence. But every now and again we do. And if you're like me, you've had an experience where you maybe made some bad choices at a family barbecue, ate some chicken that maybe was sitting in the sun too long, and then spent the whole weekend in the bathroom hugging the bowl and thinking about anti-salmonella and how maybe that wasn't such a good idea to eat that chicken. Anyways, that's one of those examples of when our microbes go rogue on us and an uninvited guest comes to the party. But for the most part, the bacteria inside us aren't just camping out. They're actually fulfilling really important jobs. Some break down insoluble dietary fiber into component parts that we can use for energy. Others produce vitamins that we need to do the work of our body. Some prevent colonization by other pathogens, and they even help us develop our own immune system. But really, who's who in the neighborhood? I don't really know. How about you? Until fairly recently, we had a ridiculously hard time really accessing information about who is living in this ever-changing microbial population. In part, this is because our digestive tract is long. It's about 25 inches long, and from 1 inch to the next along our intestine, the environment, the living conditions are completely different. Just like different neighborhoods attract and support different residents, people who live in Greenwich Village might not be the same people who live in, the suburbs of Connecticut. Well, the same is true for inside our intestines. So we could go around and do a survey of every inch of our intestine, but that's really hard to do. Sampling every block, that's just not a really great way to know everything in our intestinal city. So more commonly, what we have done is recognized that our intestinal tract is also a giant plumbing system, and that these little tiny creatures living inside us, they don't have arms, so they can't grab onto the pipes when things flush through. And so poor little research assistants around the world in different labs are caught up doing the job of collecting fecal matter and identifying what's living inside them. So those two different techniques, whether that's a biopsy of different places along the intestine or taking the excrement thereof, produces some sort of biomass. But now we need to figure out what's going on in here, who's living in here. We can't very well call roll and say, hey bacteria, can you tell me who you are and you are and you are? So we plate them. That's been the gold standard in trying to identify bacteria for a very, very long time. Except the problem with this is that whether from feces or a biopsy, about 75% or 70 or so percent of our intestinal inhabitants are afraid of oxygen, and so they won't grow on a plate. So this kind of hampered research into the gut biome for a very long time, is that we just really didn't know how to get a good understanding of what was living in there, who they were and what they were doing, and where they were from. But more recently, there's been really great scientific advances in DNA and ribosomal RNA sequencing, and this is allowing us to not only identify, but quantify a lot of the organisms living in our microbiome through their genetic markers. So we can generally now recognize the usual suspects in our guts, or at least their fingerprints. And that's great. Not a perfect system, because if we don't know who's there, it's really hard to say what it is. On the flip side, we have this whole scientific exploration into metagenomics, where we actually take all of the molecules in that, all the genetic molecules and gene fragments from this microbiome, chop them all up, and in mass computing start to identify different gene fragments that are in that sample. Now, the problem with these metagenomic approaches is that just because the genes are there doesn't mean that they're being expressed. So what we learn a little bit more is what potential actions could be taken by the microbiome in our system, but not necessarily what actually happens with it. Anyhow, all of this is to say that the world of technique has really opened up a world of research into microbiome. And this is really important for us because even new research that's coming down the pipe is showing us that knowing who's living in our ecosystem, in our guts, is actually having a really big impact in our understanding of how not only our nutritional health is being impacted by these little buddies living in our systems, but also other parts of our health that I certainly never imagined was being impacted by little microorganisms that I can't see. So let's give you some examples of some of the research that's coming down the pipe. Well, first and foremost, did you know that your gut and your brain are talking to each other like all the time? And it's not just the tummy grumbling that happens when you're hungry for a sandwich. They're talking to each other through this party line that's being dubbed the gut-brain axis. The GBA, gut-brain axis, is the communication pathways going back and forth between our gut and brain. And what's really shocking to me is that it makes sense that the gut could talk to the cognitive parts of our brain, right? We need to know if we're hungry, we need to know if we're full. That makes perfect sense. What I didn't expect is that this pathway is actually talking a great deal to our emotional centers in our brain. And so that in itself is sort of mind-blowing. And then beyond that, some of the research that we're finding out is that it's not just the communication that's happening, it's who is influencing the message that's going back and forth. So researchers are starting to look at the microbial contents of the guts of different people, especially experiencing different kinds of mood disorder or other types of mental health challenges. So there are studies that are showing that if you change the microbiota in a person with autism, you can actually change the severity of their symptoms. That's mind-blowing to me. We're seeing similar data on anxiety, stress, memory, other depressive type disorders, and even post-traumatic stress disorder. I mean, this is huge. Mental health is a major component of the public health sphere that we're all trying to impact and ameliorate. So this, of course, is making the suggestion that if we change our gut bacteria, we can possibly change our mental health status. And this concept is really being championed by these researchers, John Cryan and Ted Dinan, with this concept that's now being dubbed psychobiotics. Psychobiotics operate on this assumption that either through diet or the consumption of pre and probiotics, you change your gut microbiome, and that's going to change your mental health. Now, this is all still fairly preliminary research, but it's exciting. It's not the only cool stuff that's going on between our understanding of our health and our microbiota, though. So there's lots of research going on over the last decade or two about how the gut's microbiome appears to hold sway over the physical features of our body as well. And one of those areas under a lot of investigation is obesity and the subsequent problems with type 2 diabetes. Now, in humans, there have been tons of studies that support the claim that there is at least a significant different microbial composition between people who are obese and people who are lean. And yeah, there's always the naysayers, and there is some controversialness in some of the data because it doesn't hold true for all parts of the intestine. But as it stands, our understanding is that at least in the lower part of our intestine, we're looking at different microbial compositions between the lean and the obese. Now, there are also really interesting research studies with infants where you take someone who's 6 months old and figure out what their microbiome looks like, and then follow them forward till they're 7 and investigate whether they fall under lean or thin categories. And what we're seeing is that those infants did have different microbial makeups as infants, and that sort of predicts what's going to happen to them later on. Now, of course, there's a bit of a chicken and the egg argument here. What happens, diet is highly influential on the gut microbiome, and diet is also influential or potentially influential on obesity risk and diabetes risk. What we're saying, though, overwhelmingly, is that there appears to be some sort of correlation, and it bears further interest. And this is also predicated on a bunch of mice studies, and I'm not going to get into the mice studies, but there is really cool stuff if you want to read about them. Tons of studies showing that if you take mice and have them existing in an environment where they are not exposed to microbes, they have a completely different obesity versus thinness risk. And if you bring in bacteria and subject them to bacteria, either from obese mice or from thin mice, then you might be able to change their outlook, and it's fascinating and mind-blowing. But we all know that what happens in mice doesn't necessarily follow through to what happens in humans. So I'm not going to spend a lot of time there, but let's just suffice it to say, well worth reading some of those studies if you're really interested, because it's thought-provoking, and it does suggest that there could be correlations in humans. So what's next, right? Studies on microbiomes and the host of potentially related ailments is really just in its infancy. And being just the tip of the iceberg as it is, this is all very exciting. Already there's a burgeoning industry in the pre and probiotic fields. There's lots of people selling products that claim to be able to influence your gut micro environment. Not all of these are well established. There isn't a lot of good study that say for sure how well they work and what the influences are, and if we can store and really in the long term influence any changes inside the guts of consumers. But the great thing about consumers is they don't really require proof. People are really happy to jump on bandwagons and make changes based on speculation and incomplete data, which is really a driving factor that makes us need to continue to do this research to ensure that people aren't doing things in the absence of information that might actually be hurting them. So that's really important. But what we're seeing, what this really tells us, is that consumers are genuinely interested. They're ready to make health changes based on these ooey little creatures that we can't even see in our bodies. And I think that's really cool. If I pick and choose my yogurt based on, which bacteria I want, how cool is that? As we continue to move forward, I think that the potential is that we're going to see things like purposeful microbial inoculation, potentially even in infants right after birth. This could be especially helpful for people who are born by cesarean section who aren't immediately exposed to the vaginal microbial experience that the rest of us get. They could lead in the future to different kinds of vaccines where we're actually purposefully inoculating people with live bacteria of different kinds because we find that they're purposefully helpful. And so there's a lot of ways to see this going forward, and all of that is really interesting. And in fact, we've already seen it in the past. For quite some time now, we've been doing fecal transplants of healthy bacteria into people who are fighting C. difficile infections and other kinds of intestinal infections that can be quite harmful, especially for the elderly. There's even this great story about this health researcher, Laura Patterson, who herself was struggling with complications of Lyme disease and really struggling health-wise. And so she decided to give herself a fecal transplant from a competitive cyclist that she was doing research with. And then within months, she herself was all of a sudden finding herself healthy enough to train harder and feel better, and eventually went on to win her own cycling championships. So, I mean, this is very, there's no proof here. It's just a really interesting way to see how people are embracing this idea that even the microbiome that comes out in your feces can be useful for someone else, and we can transfer them around. And surprisingly, the ick factor doesn't seem to be as much of a limiting factor as you might think. Here in Canada, we are actually creating fecal transplant matter in a lab instead of actually having to use it from donors, and that's very cutting edge as well. So as we go on to be able to identify more and more microbes that are helpful for our bodies or can combat or help us do certain things, then we can look at these sorts of ways of harvesting them and creating an environment that's more and more susceptible to hosting and maintaining these kinds of bacteria or other organisms inside our body. So my point is, we have no idea where this line of research is really going to go. We can see where it's going and that there's a really healthy appetite for it, though. So what does this mean in terms of nutritional assessment? Well, for dietitians and other health researchers, we have historically relied upon 4 pillars of nutritional assessment to give us data on an individual's nutritional health. They're called the ABCDs. So we're talking about anthropometric measurements that tell us about growth and size. We're talking about biochemical analysis, so using blood and serum markers to tell us about what our body is doing in terms of metabolism, what we're storing in our blood and tissues, and how, giving us a very full outlook on how our bodies are presently, looking health-wise. We have clinical assessments where we actually look in people's eyes, look at their skin, see how high they can jump, how strong they are, and determine if they are nutritionally doing well, in a very functional way. And then, of course, we have dietic studies where we're looking at what people are actually consuming, which, of course, is related to their socioeconomic status and their culture and what they have access to. And there's, a lot of factors there, especially in a public health lens, where we can try to influence what people eat and what they have access to. And so these A, B, C, and Ds have really been the tools that dietitians and other health researchers and clinicians are using to determine how someone's nutritional health is being shaped. And when used together, they can inform education and public health policies, steer our monitoring and screening, and of course help us develop specific interventions to help people. So what's missing from this current alphabet soup is this new trend towards understanding the microbiome and how it influences us. And I proffer that in the not so distant future, this will become the 5th element of our ABCs of alphabet soup and nutritional assessment, is that microbiome, whether that becomes the E in environment, talking about the gut environment, or we call it the M for the microbiome, or we give it some other fancy name, the bottom line is what's going on in our gut is influencing our health, not only nutritionally, but also all aspects of our health, and therefore may play out in the future to be a tool that we can use to do nutritional assessment work. And so I really want to thank you for talking to me and hearing about what is going on in the microbial world and talking about our little friends and foes, the organisms living within us. Thank you so much.